So welcome everyone. Well, it is great to see so many people interested in today's talk. My name is Milena Grzybowska and I'm a committee member of OsteoSIC and representing commercial sector. It is my great pleasure to introduce you to our very first Osteobites guest, Dr. Thomas Booth. Tom is bioarchaeologist and a senior research scientist working in the Ancient Genomics Laboratory at the Francis Crick Institute in London. Tom has been working for the past several years on a 1,000 ancient genomes from Great Britain project funded by the Wellcome Trust, and the project that aims to undertake a fine-scale reconstruction of human evolution by sequencing at least 1,000 human genomes. Very recently, we could read about some of the results of the lab's work in the journal Nature in an article focusing on a genomic history of early medieval Europe. Today, Tom will present a talk entitled Ancient DNA for Archaeologists, what it can and can't do and why. And we hope to get better understanding of what has changed in the ancient genetic studies and how we can contribute. So with no further ado, I will hand you over to Tom.
Thanks for that introduction, Melina, and thanks for organizing this talk. Hold on, a series of talks. Sure, start. Okay, all good. So I think it'd be useful to start with a little bit of background about me. Firstly, I originally was an archaeological scientist trained at the University of Sheffield, so my original degrees are all in archaeology, but then back in 2014, I started work on an ancient DNA project at the Natural History Museum. And really, that was my first real interaction with ancient DNA as it's understood today, after, what's often called the archaeogenetic revolution. So I learned about the ancient DNA. I've been working on ancient DNA projects ever since, having come from a background of archaeology. So hopefully that gives me a certain level of insight and certainly a certain level of sort of knowledge of the genetics on a need to know basis, whereas I don't need, and I haven't got, and I would never claim to say I have an in-depth understanding of molecular biology, but I know, I feel I know enough to understand the concepts and how it applies to archaeology. So hopefully that gives me the ability to communicate it to other people, because it is bewildering, and I doubt if I'd have been on these projects, I would have a really particularly well-rounded understanding of things. So I'm hoping that this should give you a relatively non-technical understanding of the basics, what has happened in ancient DNA, which has meant that over the last 15 years or so, it has become a much more viable prospect than it used to be, and what the applications are and kind of continue to be specifically for archaeological sites that you might be working on. But firstly, inevitably, a plug. We're still, as Melina said, we're still generating data from ancient humans from Britain and also from continental Europe as well. And we have our own research aims looking at quite broad scale questions, but for each individual that has decent DNA preservation, we can give information on genetic sex, ancestry, relationships between individuals, and potentially infectious pathogens. So feel free to get in touch if you are interested in us analyzing your samples, and we try our hardest to work with collaborators to use the data to answer your specific research questions as well. It's not just about our research questions. We want to get more out of it than simply sort of a big paper with just everyone's name done it splashed at the end. Hopefully we want to generate more papers that look specifically at results from specific sites. So please get in touch if you're if an interesting site and are interested. I mean, most of the time we'll say yes, but I mean, the worst thing is we say no, we can't. So I'm going to start just talking about some key concepts in DNA, which again, hopefully isn't too technical, but I think is useful to have an underpinning of what we're doing and the methods that we use. So, I think that this is a common theme that you can think of ancient DNA, or sorry, DNA as a sequence of letters, GCAT, representing different what are called nucleotides. And these sequence of letters, they are always opposed by a different letter. So G on one side of the double helix of DNA, the double helix of structure of DNA, is always linked with C, A is always linked with T. So these are sometimes called nucleotides or base pairs. And the human genome contains around 3 billion of these base pairs to make up the genome, organized into this double helix structure. And sequences of DNA are organized into pairs of chromosomes. So you have the DNA double helix, so those base pairs twisted around these histones, and when they're twisted around, they form these chromosomes. So in humans, we have 22 pairs of chromosomes. So each sequence of those base pairs, we have 2 copies of them in the chromosomes, 22 pairs plus the sex chromosomes, although a minority of people have different numbers of chromosomes, chromosomal aneuploidies, which I'll talk about later. So we inherit DNA from our parents. So we get for each pair of chromosome, one of our chromosomes comes from our mother, one from our father, and that chromosome that you inherit is actually recombined. So it's a combination of each parent's 2 chromosomes, which recombine into one chromosome. So on each chromosome, you've got a mixture of your parents' DNA on each chromosomes, and you inherit one of those from each of your parents. So mixtures of ancestries through recombination. I thought I'd just mention genes here quickly because I think it's often a misused term and misunderstood. So genes specifically don't necessarily refer to the entire genetic sequence. It's specifically sequence of nucleotides of these base pairs that we know encode for proteins. So we can say directly functional and produce proteins, and these can be distributed. They can be these variants that are on genes can be distributed across chromosomes, and different versions of genes, as you may already know, are called alleles. But most of the human genome is not genes. It doesn't directly code for proteins. It doesn't have a direct functional effect. But it may be that many of these variants are neutral. Many of these variants can sometimes regulate or interact with genes and control what they're doing. But genes specifically refers to these functional parts of the genome and not the majority of the genome, which is often what we're investigating. And you'll often come across this term a lot in genetics and ancient DNA, SNPs, or single nucleotide polymorphisms. So you think of these sequence of GCATs. A single nucleotide polymorphism is a position on the gene on one of the cGNAs and Ts that commonly varies between individuals or populations. So technically, every variant in the genome has the potential to be a SNP because it has the potential to vary. But in reality, there's a certain number of SNPs that commonly vary between people, and there are around between individuals or groups, and there's about tens of millions of these SNPs out of 3 billion base pairs. So the vast majority of these positions in humans, over 99%, are essentially the same. And it's only these tens of millions of SNPs that vary on the DNA sequence that vary between people. So even though it's true to say that most humans are genetically, over 90% similar based on this model, that still gives you tens of millions of independent points of data. So it's still an extremely powerful data source and allows for quite intensive analysis, despite only being a tiny proportion of variation across the genome. And some SNPs are close to or within genes with either a direct or indirect regulatory effect, but most are neutral. And like genes, different variants of SNPs, slightly confusingly, are also called alleles. So you've got a certain position in the genome that varies between 3 different people, and then each type of that, whether it's A, G, T on that particular spot, those are different alleles of that spot, just like different versions of genes, but genes are a much broader phenomenon. But we tend to deal in these SNPs, these single variants. When does ancient DNA become ancient? Essentially, not really when it's ancient. Really, ancient DNA is defined by the fact that it's fragmented. So very quickly after death, your DNA unravels and rather than being this long sequence of GCAs and Ts, it gets broken up into little pieces and continues to be broken up through time. After these fragments are broken up, chemical reactions with the ends are sort of exposed to chemical damage. So a chemical reaction on the ends produces what's called the C to T damage, where cytosine is chemically altered into thymine. Yeah, that's right. That accumulates on the ends of these fragments, and this damage progresses in association with time, temperature, and precipitation. So you could actually have DNA that's not that old. If it shows these factors, you could technically classify it as ancient DNA. So moving on to what has changed in ancient DNA and why we're suddenly able to say a lot more and say so pretty sensational things, seemingly quite robustly. So the older methods before around 2010 or so involved polymerase chain reaction and Sanger sequencing of small aspects of the genome. So you would demineralize the sample, extract the DNA. And the problem was that sequencing, the Sanger sequencing that was used back then was very expensive. You could only really afford to sequence maybe hundreds to thousands of base pairs, nucleotides that you're interested in. So you would target a particular part of the genome that you're interested in to answer your question, whether it was to do with, say, lactase persistence, whether people could digest milk into adulthood, or whether it was to do something to do with ancestry. You would find genetic sequences that were a tiny proportion of the genome, like I said, hundreds of thousands of base pairs, and you use these things called primers to enrich and clone your enrich your DNA for those sequences in particular using this polymerase chain reaction, which is essentially, I won't get into the technical details, but it's essentially the cloning of the DNA sequences, these specific sequences, and then you would sequence these using Sanger sequencing, which was very expensive. And while they were to some extent informative, these sorts of studies, they were relatively tiny, although informative portions of the genomes and done very strategically. And one of the main issues was that it was very difficult to control for contamination. You had no idea whether the sequence that you were enriching was genuinely from the ancient sample that you were looking at, or was just a bit of skin that had dropped off you or from someone that had handled the remains. There was no way of distinguishing between the two. There could be some genuine results from that period, but it's difficult to distinguish between what was genuine and what wasn't. The main thing that's changed ancient DNA has been the advent of next generation sequencing. And the main point of this is that it has massively reduced the cost of sequencing. So this meant that you could start to sequence millions of DNA sequences economically rather than just these hundreds to sort of few sequences that were these little bits of DNA. And this was also accompanied by advances in extraction techniques, which allowed optimal acquisition of very small fragments of DNA, which comprises the majority of ancient DNA samples, because all of the DNA has been chopped up. So what that means is that rather than getting these tiny bits of DNA, we were getting sequencing DNA, sort of millions, hundreds of thousands to millions of sequences distributed across the entire genome. We didn't have to target a specific part. We just got bits and pieces from everywhere. So much, several orders of magnitude times richer data. And these so the current laboratory methods again involve demineralization and lysis to release the DNA. You extract the DNA, which produces these laboratory products called extracts. And then those extracts of DNA are produced into what are called next generation sequencing libraries. They're called libraries because essentially what you're doing is you have these little bits of nonsense DNA called indexes that you know the sequence of, and you attach them to the ends of the DNA sequences that are in your sample. And what that means is that you can mix lots of samples together to sequence on a sequencer, and then you can pull them out again by sample based on these little indexes that you've attached to the ends of the DNA sequences. So again, this all adds to the sort of economy of being able to sequence DNA now, which allows for rich ancient DNA sampling. So yeah, as I mentioned, I mean the thing to so when you have the bone powder in these lab processes, the DNA extracts are finite. You eventually run out, but the libraries are produced through this PCR method where you polymerase chain reaction where you're cloning DNA sequences. Essentially, if a DNA library, that library is technically immortal. If you start running out of the library, you can just clone more and more and more. So it's potentially a totally renewable resource to some extent. And then sequencing produces reads of fragmented sequences. And then the way that you pull out the human DNAs, we have a reference sequence, and essentially there are algorithms and software that find these little sequences of DNA and match them to the reference sequence to build up the where all these sequences fit on the human sequence. So it's a bit like that all the sequences are bits of a jigsaw, and then this human reference sequence is the picture on the jigsaw, and then you use the computer algorithm uses the reference sequence to build it up to build the genome that you have. And then often, well, almost in all cases, the data is uploaded to publicly accessible repositories on publication. So again, compared to a lot of other analyses, DNA has a quite strong legacy in that the data is there for people to return to. If people need to resequence, get more data from a sample, they can always go back to the library, which they can sequence, and libraries can be sort of maintained in perpetuity. If for whatever reason they have to go back to the extract, there's the extract potential, or they will eventually run out. But you have all these stages that people can go back to before they have to sample a bone. So the kind of point is that a lot of the sampling that's going on for DNA now, in theory and hopefully practically as well, it sort of negates the need to have to go back to resample a bone in the future, because kind of producing these sequences and producing these laboratory products should mean that they persist and can be gone back to over and over again. So kind of the main standard version of sequencing is this next generation shotgun sequencing, and it's called shotgun sequencing because each time you're sequencing, you're actually getting sequences, a scattershot set of sequences distributed from all over the genome. So you're not actually selecting what sequences you're getting back. It's just random. But the more and more you sequence, the more and more you get back. So the better coverage you have of genomes. So it's called, when you do that, it's called whole genome data. But this doesn't mean that you have the whole. It's a bit misleading. The whole of a genome. You don't have all of the genetic information from an ancient individual. I don't think there's any ancient individual where you have the complete entire thing. I mean, it was only just recently where technically we had the map of an entire modern human genome. I mean, it sort of depends how you classify it. But whole genome data just means you have shotgun data from distributed across the whole genome. It doesn't mean that you have the entire genetic information from that individual. But most of the time, we don't need an entire sequence to answer most of the questions that we're working with. And the main thing that this analysis does, or one of the main benefits of the analysis of this next generation shotgun sequencing, is it's allowed for the development of methods that authenticate ancient sequences, allow us to assess contamination. So we can control for and identify and control for contamination, which wasn't possible in the past. So in terms of looking at preservation and assessing contamination, one of the main things we look at is the endogenous content of the sample. So this is the proportion of the DNA sequences that we get back that align to the human genome. If we're looking for human DNA, aligned to the human genome. Obviously, if you're looking for pig DNA, you look for what aligns to the pig genome. And initially, when next generation sequencing was first applied to ancient samples, the big problem was that the vast majority of sequences that you were getting back were from microorganisms that lived in the soil, and a tiny proportion of it would be from the human itself, which is understandable given they've been buried in the ground for centuries. And this means, I mean, a poor endogenous content means it becomes uneconomic to sequence remains, because if you're sequencing is one of the most, well, probably the most expensive part of the process, even now when sequencing costs are still going down. So if all of 90%, 99% of the sequences that you're getting back aren't from the organism that you're interested in, it just becomes uneconomical to carry on sequencing because you're just not getting enough information back. Again, this is starting to become the threshold for what is uneconomical increases is sort of going down as sequencing costs are going down as well. But at least particularly in the early stages, sort of things with 1% DNA were very difficult to get a decent amount of information of. And one of the ways this was resolved was that people realized that sampling of teeth and petrous portions of the temporal bone, so the temporal bone around your on the side of your head, around your ear canal, as well as auditory ossicles, preserve DNA much better than other parts of the skeleton that people have looked at so far, really rich in endogenous DNA. So it's not a magic bullet in that sometimes there can be zero or very low amounts of ancient DNA in a petrous or an ear ossicle or a tooth. But so particularly the ear ossicles and the petrous, you can get up to something like 90% endogenous content. It can be really rich sources of DNA. So that's the targeting of these particular bones help get around that aspect. Yeah, so auditory ossicles, so the tiny inner bones of the inner ear, temporal bones and teeth. We've had recent successes, and other labs have had recent successes with phalanges and tali at a push where there aren't any cranial remains available. I mean, they don't work anywhere near as often as other bones, but where you have no cranial bones, they seem to be the best ones to try. But I'll talk a bit about pathogen DNA, disease DNA. It seems to more often turn up in teeth. It occasionally turns up in the temporal bones and ossicles, but very much more often turns up in the teeth because it's much more proximate to a blood supply where we assume that most of the pathogenic bacteria is hanging out, whereas in the auditory ossicles and temporal bones, they don't have as much of a blood supply, so there's not as much of a representation of the bacteria in those bones. The main way of authenticating DNA as genuinely ancient is through the C to T damage that I mentioned earlier. So looking at the ends of the damage on the ends of the fragments of DNA, which shows that it's genuinely ancient. So any recent DNA shows none of this damage at all. You can also gauge contamination by looking at the mitochondrial DNA. So I talked about the diploid genome, human genome, that you have two sets of chromosomes. Well, your mitochondrial DNA is kind of a separate bit of DNA, which is just a single strand. It's not sort of organized into a pair of chromosomes. So mitochondrial DNA, you know that you should only have sequences that are what are called homozygous. All your sequences positions should be the same because you don't have an opposite side, an opposite chromosome that could be different. So by looking at to what extent you have different alleles represented at the same position on the mitochondrial DNA means that you can assess if you're having lots of different positions, that suggests that there's two mitochondrial genomes in there. So by looking at the proportion of positions where it looks like you've got two mitochondrial genomes, that gives you a sense that it has to be somebody else's mitochondrial genome. So you can assess what percentage sort of contaminated that is. Because genetic males only have one X chromosome, an X and a Y, you can do the same thing for the X chromosome in males to assess contamination. Have a look because the positions on the X chromosome should all be the same, or should be coming back at the sequences from the X chromosome should all be the same because in males because they only have one X chromosome, and if it's coming back as two different positions in certain position, that has to be contamination from a different person. There's also this idea of complexity, which when preservation, you don't necessarily have the whole genome, all of the genetic information from an individual within your sample necessarily. There's often a kind of plateau to how much unique information that you can get out of it as a proportion of their overall genome. And this is represented by complexity. So this is the proportion of your sample that's made up of unique DNA sequences rather than duplicates produced by these PCR polymerase chain reaction methods. So the polymerase chain reaction methods, as I mentioned, is essentially clones the DNA up to a point that gets it to a high enough level that you can sequence it. And what that means is that certain sequences that you have get cloned several times, but they are not actually adding any more new information because it's just the same sequences again and again. So the complexity is the unique DNA sequences. So and through that you can assess there's a certain plateau where this percentage of the genome is covered in this sample, and no matter how much more you sequence it, you're not going to get any more. So using this random shotgun approach. So for DNA libraries that have very low complexity, it's very difficult to get an adequate level of unique information, usually as well, because they usually have a low endogenous content as well. So in terms of thinking about the anxieties about contamination in the past, particularly with old PCR methods, we've sort of, to some extent, moved past that. I want to be careful because it's still something that ancient DNA researchers are very concerned about. And every sample is contaminated to some degree. But how much of a problem that is depends on the level of contamination and DNA preservation. So most of the time, contamination only becomes a serious problem if the DNA preservation is low. So kind of every sample has a baseline level of contamination. And if your sample is particularly poorly preserved, what happens is that your level of genuine ancient DNA is at about the same level of that contamination if it's poor, so it makes it very difficult. But most of the time, or at least in cases where the DNA is decently preserved, the signal that there's so much more of the DNA from the target individual that the kind of contamination that's there the small bit of contamination there makes no difference. So it's very much dependent most of the time on the preservation of the DNA in there rather than contamination being a major problem in of itself. So in most cases, it's quite a trivial issue. And because I mean, we've sequenced, and this is true of bones that have been freshly excavated from the ground, but also legacy collections that have been around for kicking around for a century or so that have been extensively handled. You know, we've looked at both kinds, and there's no real, as far as we can see, elevated levels of contamination in those bones that have been kicking around for a while compared to those ones that have been freshly excavated. So, while contamination remains a concern, most of the time from places where we expect DNA preservation to be decent, it's a relatively trivial problem. The fact that we can control for it helps as well. So, there's the effective ways of detecting contamination, the low impact of contaminating sequences in general, largely eliminates the need for, say, excessive PPI on site when you're excavating things. I think that the days of thinking that we have to have like hazmat suits, gloves, masks to excavate anything that we want to put forward for DNA analysis are kind of behind us. Sort of desirable things, but again, not essential. You know, we've sequenced bones that have for sure been extensively handled and should be covered in DNA from people that have touched them without gloves over and over again. It's been fine. I mean, a desirable thing would be potentially sieving of soil around the skull to catch osteoarchaeologists, which are particularly rich sources of human DNA. Gloved handling of possible good sources of DNA like crania, teeth, or osteoarchaeologists. Gloved handling with gloves, even. Sorry, tautology there. At the post-ex stage, a big thing might be changing gloves if possible. Again, this depends how practical it is because it doesn't seem to make that much difference, but it might be something that's useful to do. Changing gloves if you want to be hyper cautious, changing gloves between each skeleton because it's easy to detect contamination that comes from a modern source. It's much harder if something's been cross-contaminated with an ancient source. So if you're moving between samples with the same pair of gloves, you could potentially be cross-contaminating ancient with ancient, which is more difficult to pick apart. So again, yeah, if it's at all practical, changing gloves between skeletons I wouldn't necessarily say when you're just doing generic human osteological assessment, but perhaps if you know you're going to be handling the parts that are good for DNA, like the petroses or the osteoarchaeologists or the teeth, then it might be good. But again, we've sampled lots of skeletons where people haven't done this and it's been fine. So again, it's not a supremely sort of urgent thing to do. It would be good if you had the time, if people could search for auditory. I know I go on about auditory oscills a lot, but I can't emphasize enough how good they are and how much easier it makes our lives when we have auditory oscills in lots of ways. Looking for them and keeping an eye out for them when you're cleaning the bones. But if not, it's actually better for you to not clean out the auditory canal at all and leave the sediment there because the sediment is often holding in the auditory ossicles. And we find auditory ossicles much more often in ear canals where the sediment has just been sat in there holding them in than when they've been cleared out. Now, it's not to say that you can't clean skeletons at all. That's fine. Just be careful around the auditory canals. So I was going to talk a little bit about this sort of other aspect to ancient DNA laboratory methods, targeted in solution capture arrays. And this is essentially where, particularly at the beginning, where sequencing was more expensive and you had a lot of samples that weren't very well preserved. Some ancient DNA labs, particularly in Harvard and Max Planck Institute, developed these Bates primers to enrich samples for these predefined human sequences. So they put together a list of 1.24 million sites, so these SNPs on the genome, that they knew that if they had those as a bare or as a proportion of those as a bare minimum, they'll be able to answer most of the questions that they wanted to answer about ancestry, genetic sex, relationships between individuals. So what this does is essentially just enrich the samples for these 1,240,000 SNPs and essentially gets rid of everything else. So you get rid of all of and so this means samples are essentially 100% human DNA and gets rid of everything else, which means makes them much more efficient for sequencing because you're sequencing just the human reads. The downside to this is that you're also getting rid of the vast majority of human sequences. So any sequence or variants that aren't on this 1,240,000 panel get washed out as well. I mean, for most of the time, it's fine, and that's particularly for the kind of things that archaeologists are interested in terms of ancestry, genetic sex, again, relationships. Getting a proportion of those SNPs is fine and enough to look at those things, but it has downsides in other ways. And up until now, most ancient human DNA has been generated this way because the benefit of it as well is that it means that each time each lab is getting the same set of sequences, and particularly when a sample is poorly preserved, enriching them for these variants means it's much more economical. You do many more samples than you would be able to just by sequencing them straight away using this shotgun approach, where many fewer sample, a much smaller proportion of your sample is going to be human DNA. But I mean, the downside is that it's only a fraction of a whole genome. And with the loss of the unidentified human sequences, there's potential evidence for some ascertainment bias, and the data legacy is poorer. If you find later on that there's a really important genetic variant that you really want to investigate for some evolutionary reason that wasn't on that panel, then it won't be there in any of this data that's published. And up until recently, it was really the only economical way of getting adequate information from poorly preserved samples. But this is rapidly changing. I mean, again, up until recently, kind of a mixed shotgun capture approach is optimal, where for samples that are well preserved, you just shotgun them. For samples that are poorly preserved, use a capture approach because that's the only really economical or way of getting a minimal amount, a sort of minimum amount of information out of a sample. But what's happening more recently is that because the sequencing costs are continuing to drop, it's getting to the point that even with poorly preserved samples, it's becoming economical and it has a better legacy value just to shotgun sequence them, so you're getting all of the information out of the sample rather than this sort of refined mix of variants. So I think people are rapidly moving towards just a whole genome shotgun sequencing of everything, even if it is poorly preserved. And this is better because, again, the data legacy is poorer. And the good thing is that even those libraries that have been subject to this targeted capture array in the past, people can just go back to and just shotgun sequence them. So it's not like anything's been lost with the fact that people have applied these laboratory methods in the past, because you can just go back to the libraries and shotgun sequence them when it becomes economical to do so. So the first thing that's often done in DNA laboratories is a shallow sequencing run where you generate a little bit of data from each library to see how well preserved the DNA is, looking at these metrics of damage, endogenous content, and complexity. And then from that, you decide those metrics that you generate, preservation metrics, you decide whether you can sort of shotgun sequence something, you have to put it on a capture array, or whether it fails. Although the kind of level at which a sample fails is slightly arbitrary, and it's possible that in the future, again, as sequencing costs go down, people will be able to return to samples that failed in the past and actually get something out of them, because all those laboratory products will still be available. And then samples that pass the screening stage put forward to deeper what's called production sequencing, either production sequencing of targeted captured libraries, or just the libraries without capture, so just whole genome shotgun sequencing. And this produces a measure called coverage, which is essentially a measure of data quality. So if you think about that, in this image, you have the reference sequences, and you have lots of different sequences that you've generated that cover a particular position in the genome. The coverage is the average number of unique, not these duplicate sequences, unique sequences that cover these positions. And it's measured in 1x, so 1x, 2x, 3x. So if your genome is 1x, then it means that each of these positions is covered at least once. When you're using a targeted capture, usually the coverage refers to, of those 1,240,000 variants, what average coverage have you got on each variant. But it can be quite variable over the genome, so it's very much an average in that, because of the way that different sequences preserve and sequencing happens, and how easy certain parts of the genome are to sequence compared to others. You might have sort of parts of the genome that are 0x, but other parts that are 10, 20x, and then that averages out to a certain level. So bear in mind that doesn't give you a sense of, it's not a uniform thing across the whole genome. There's parts of the genome that will be really well represented, and there's parts of genome that are poorly represented for each ancient individual. And the coverage is important because of thinking about the diploid human genome. So many analyses, because if you think about it, that when you sequence, so at a particular position, because you have two sets of chromosomes, if that position is heterozygous, so on each chromosome you have a different
variant at that position. If you sequence it once, you'll get one of them, but you're not sure of what the other position, whether the other position is the same or it could be different. So it could be heterozygous. So if you have 2x coverage, you've got another sequence covering that position. If it's the same position, there's kind of a 50% chance that is the same. It's 50% chance that, sorry, it's more like 75% chance it's the same. 75% is a different sequence. And if you've got kind of 10 sequences coverage, you'll be 10 sequences covering that position, and they're all the same. You'll be confident that position is homozygous, that on both chromosomes it's the same variant. Whereas if it's like 50-50, one variant and the other, you know that they're probably heterozygous at that position, that you've got two different variants on each chromosome. So you need that high coverage to be able to look at what the diploid genome, that both variants on each chromosome. What tends to happen is that you need really high, decent quality genetic information to be able to do that. What often happens is that studies treat the data they get back as haploids. That just means one set of chromosomes rather than pairs of chromosomes. And that works fine for looking at genetic sex, broad ancestry and relations between individuals, and doesn't require you having to know which parts of the genome have different variants on different chromosomes, or heterozygous at those sites rather than homozygous. But when you're getting to more intense questions like looking at phenotype, or looking at selection, and looking at kind of disease risk and things like that, inbreeding population size, very fine scale ancestry, you need to know what the positions on both chromosomes are. So you need very high coverage, very high quality genomes to be able to do that. So getting into the meat of things in terms of what we can use this to say, I mean, I've alluded to before. Firstly, genetic karyotypic sex. This is usually calculated through just looking at the number of reads that align with the X and Y chromosome. With broadly speaking, for males, it should be genetic males, it should be kind of 50-50, whereas females, we shouldn't have many reads aligning to the Y chromosome apart from what we get from any contamination. And this is often possible. This is one of the basic things that we can do in quite poorly preserved samples. So samples for which we can do anything else with, we can usually get genetic sex for. So I'd say around 90% of the samples that we look at, we can at least get a genetic sex estimate. So this is obviously helpful in instances where sexing is uncertain because of preservation, or indeterminable because it's a subadult, or they just show the skeleton shows a mix of features that makes it difficult to sex. We also pick up on karyotypic aneuploidies, including sex aneuploidies. So this was a study that my colleague Kakia Anastasia published last year, essentially developing a method for distinguishing between these different sex chromosome aneuploidies, as well as other aneuploidies like trisomy 21, so Down syndrome. And you can see that sort of in this graph, the bulk of samples fall in XX, so the yellow ones, and the XY, these sort of burgundy coloured ones. But you get odd individuals that fall in different places on the plot in terms of their ratios of sequences of X to Y, with XYY, which I think is Jacob's syndrome, and XXY, Klinefelter's, and we want X0, which is Turner's syndrome. So this allows for potentially, because I mean, some people have these intersex disorders and there's no real visible physical effects at all, but some people there's quite physical effects, and often because of the way in which the sex chromosomes regulate sexual characteristics, it can often mean that people who have these disorders, sort of their gender or their gender identity, or how they think of their bodies, influenced by these disorders. So it can potentially be a new route for exploring intersection between sex and gender in the past. I mean, like I said, it's difficult to say. I mean, a lot of the samples that have been looked at so far that have come back with these disorders, there's nothing to indicate that they identify with a different that their gender was different to what their sex was. But it might be one avenue in which we can explore this in other samples. Looking at genetic relatedness, so early method was based on allele sharing. So where you could detect first degree relatives. So if your first degree relative, you'll share 50% of your alleles, so the variants on those SNPs, you'll share 50% with first degree relatives, 12 to 25% with second and third degree relatives. And you can combine these sort of degree relative scores with radiocarbon dates, uniparental haplogroups. So these are bits of DNA from your father and your mother. So if people share a uniparental haplogroup, they share a maternal or a paternal lineage, and sort of stratigraphy as well to work out relationships. And this has been really fruitful. In fact, most ancient DNA studies are essentially just huge genealogies that people have managed to work out from patterns of genetic relatedness. And what's increasingly happening now are you're looking at these questions using these identity by descent segment sharing methods. So an identity by descent segment of DNA is a bit of DNA that's, like I said, a segment of DNA that's inherited together. So if you look at this figure, you can see that this red line, these sort of couple with the red, these sort of represent chromosomes. These lines are chromosome from a mother, chromosome from a father. You see that the next generation, one person's inherited essentially this entire chromosome from their parent. This one's inherited half of it. Then the next generation, they've inherited half of it. They've inherited half of that previous one, which means that this proportion of the genome here that these individuals share has come directly from that joint ancestor at the top. So these segments of DNA essentially represent shared genetic ancestors. And in order to get these to detect these identity by descent segments, to look at shared genetic ancestors, you need quite high quality DNA. But once you have them, it helps clarify close genetic relationships. So rather than just being able to say that two individuals were first degree relatives, you can usually say whether they were siblings or parent child, because the way that the IBD segments are dispersed amongst siblings or parents is different. But it also allows you to identify much more distant relationships, so 6th degree relatives and beyond. So that could be if two people share a segment that's equivalent to 6th degree. So basically, each recombination event that segment gets that you can compare to an ancient individual gets smaller. So by looking at the size of that segment, that relates to how far in the past that shared ancestor of those two individuals was. So that's how you generate the generational time. So you can identify people who share distant genetic relatives. So so potentially quite distant cousins who will share a distant genetic ancestor, but also potentially direct relationships between sort of one person who is the direct ancestor of another, but also kind of everything in between that. So you might have lots of avuncular relatives through which you will share DNA segments through complicated relationships. So often when you get back beyond this sort of 6th degree back to these 6th degree relationships, it's hard to clarify exactly what the relationship is, but it can help. For instance, you could use this potentially to identify long-lived familial burial sites, but probably the use of these sort of IBD sharing networks is on a kind of broader scale, where it can help you look at the nature of a cemetery population. So if you expect your cemetery population to be quite genetically insular, you would expect that even if you're not necessarily picking up, particularly if it's only a part of a cemetery, you might not necessarily be picking up lots of close genetic relatives, but you would expect that quite a few of them would be distant relatives of each other. However, if you had a kind of cemetery that was quite a mixed population of people coming and going, or a population that was quite large, you would expect to see very few distant relatives within that group. And this can be useful in places where cemeteries where broadly the ancestry of the people in the cemeteries was the same, but where there was kind of mobility within people of similar ancestries. So you can see these different dynamics within different cemeteries. And also you can look at these longer term networks of regions of biological interactions between cemeteries as well. So you can get these networks of IBD sharing that give you a sense of reproductive interactions between different groups, like this study here by Lara Cassidy that came out recently on Iron Age Britain, where the colours represent different networks of IBD sharing between different sites. So you have, say, the purple ones in Cornwall, people having children primarily amongst each other in Cornwall, but also across to Brittany as well. So you have this particular network of interaction between Cornwall and Brittany, and then in the red ones in Dorset, and then some in south central, southeastern Britain amongst each other, but also with people from continental Europe. So this gives you more of a sense of long-term networks of biological interaction, which can help you understand sort of potentially zones of at least biological interaction. Sorry, I'm overgoing. I might have to rush through this last bit, but moving on to genetic ancestry. Genetic ancestry, when we talk about genetic ancestry, it's kind of one of the more complicated aspects of DNA generally, and probably doesn't necessarily relate directly to a kind of a popular understanding of ancestry. But generally, you can think of it of representing shared recent ancestry, representing shared genetic ancestors up through the last 300 years or so. And one of the ways in which this is looked at is through allele sharing. So if you think about what I was talking about IBD segments, essentially all your DNA is made up of these IBD segments that represent your different genetic ancestors. And at the allele level, that means that people who have a similar pool of genetic ancestors will have more alleles in common than people who share different pools of genetic ancestors. And because humans reproduce non-randomly because of geography, topography, culture, you get structure in that sharing of genetic ancestors. So some people will share more genetic ancestors than others because they live closer to one another, because they have linguistic affinity or cultural affinity, or because there's a big mountain between them and the people next to them. And this produces the structures as genetic ancestry. But it's important to note that the structures that we pick up in genetic ancestry exist at all resolutions. So you could follow these structures down all the way to the level of sort of a family, and then all the way up to kind of a geographical sort of broad geographies like continents. So there's no resolution at which that ancestry is the ancestry. It's whatever resolution of ancestry is pertinent to your questions. Then newer methods like what Milena mentioned earlier, Leo Spiedel's paper looking at early medieval Europe, use this haplotype specifically. So you and one individual could share an allele because you have a shared ancestor in the past, or it could just be chance. Whereas these longer sequences of DNA that you understand the genealogical relationships between, that's less likely to happen by chance. And also these IBD segments that people share, you can sort of date when they occurred. So you can restrict your analysis to just looking at these segments that occurred relatively recently. So you're only looking at process that happened in the recent past, which allows you to distinguish between the fine scale ancestry of, say, people who inhabit people who are genetically quite similar to one another. So for instance, the people who largely inhabited Europe during the early medieval period. So it allows much more fine scale understanding of ancestry. It also requires slightly better quality DNA than you would normally get, but again, it allows for much more fine scale reconstruction of ancestry. And because of this genetic variation representing the individual and their ancestors, it means that it's quite robust in that you're not only getting a signature of an individual, but their population of ancestors as well. So hundreds of thousands of people whose remains might not have survived into the record. So a lot of the time, sample size is less of an issue, but sampling bias might be a problem if you're having groups of people living alongside each other that don't have children with each other very often, so producing quite a lot of structure in the data. And particularly if one population is doing things with their dead that leaves an archaeological record and the other one isn't. So it's always important in these studies to bear in mind that we study cemetery populations and not living populations, and extrapolating what you see in the cemetery to living populations always involves a series of assumptions about them representing a population. I think certainly a lot of the question that I get, and probably a lot of archaeologists get when they're on sites, is figuring out whether the people that they're digging from cemeteries have any living descendants. But the problem with this is it kind of misunderstands the relationship and whether you can test that with DNA. But this sort of misunderstands the relationship a little bit between, depending on how old the cemetery is, genealogical ancestors and your genetic ancestry. So as you go further back in the past, your number of ancestors is increasing exponentially. But while your genome is a rich source of information, it's also finite, and you can only have a certain number of ancestors represented in your genome. Eventually get to the point where you have more ancestors than will fit into your genetics. So what happens is that past a certain number of generations, your certain genetic ancestors start to drop out of your genome. So even though they're your genealogical ancestor, you've inherited no DNA from them. And that starts happening back from around 10 generations back or so. So if you look at this graph here, this is 17 generations back. And the x-axis is distribution of ancestors over space, and the blue is the ancestors from whom you've inherited DNA from, and the red is the number of ancestors that you've inherited no DNA from. So you can see very quickly it gets to the point where people who are genealogical ancestors you've not received any DNA from. So probability means that you're going to have more ancestors that are directly relevant to your population history than are kind of more dispersed across well, the world to some extent. So genetically, you're more likely to inherit DNA from them, but generally speaking, your genealogical ancestry is sort of legion even a few generations back. So this means that you can't really connect living people meaningfully to ancient ancestors, because it could be that thousands, tens of thousands of people alive today are the descendant of an ancient person, but only a tiny proportion of those people will then have inherited any DNA from them. So you can't really use DNA to connect people to ancient populations in a meaningful way past a certain point. And when people are connected, if people do share a bit of DNA with an ancient person, then it's interesting, it's cool, but it doesn't really mean anything, because plenty of other people have exactly the same level of relationship, and just by chance they won't have inherited any DNA from them. So if anyone asks you, can we see if there are any living descendants, you can say it's not really possible. Sorry, I'm really cutting things fine here. So there are different ways of thinking about migration. So one of the most famous, really, coming out of ancient DNA studies is the population level of migration. And migration in genetics here is really defined very broadly, and people moving from one area to another and having children. And the having children part is mostly important because at a population level, at least, we don't see it unless the people moving are having children. And ancestry change at a population level over time is often called a population replacement. But I mean, all that means is a shift in ancestry. It doesn't imply anything about the mechanism of genetic change, and that indicates migration, that people are moving to a new place and having children. It does not directly equate, no matter what some people might say, to invasion or wipeout. Various mechanisms could be responsible. So any factors which means that people carrying any migrant-derived ancestors are more likely to have children who have children, etc. Or particularly over centuries, even if it's just a slight advantage. For instance, if there's a slight infant mortality, if people with ancestries derived from migrants have a kind of lower level of infant mortality over the long term, that can create a big difference in genetic ancestry. So while we can say that these genetic changes happened, usually because that we have modern genomes to compare them to, so any genetic change we can see in the past, we can look at modern genomes to see whether that's been pervasive and that was kind of permanent, that change. So we can say these changes happened. How they happen is something that genetics by itself can't answer and needs to be integrated better with the archaeological information. We can also look at migration at an individual level. So if a person has ancestry that's distinguishable from others on the same site or the same region, but importantly, if that happens, that they don't have the same shared recent ancestry with people on the same site, we can't strictly identify them as a first generation migrant, only in combination with other techniques, because it could be that they're second generation or a descendant of a community of migrants comprising a minority of a population that's maybe formed an enclave. But comparison with other populations can help find a likely source of ancestry, but this is a very intense process in that you are likely to have the exact source, so you have to go through some level of triangulation where you're comparing it to people that are maybe in the right area that you think they might have come from, but at the wrong time, or in the right time, but the wrong area. So by comparing them, you come up with a plausible idea of where their ancestry is likely to come from, but it's likely you have an absolute true source. And in some cases, like for instance, Rome, it could be that ancestry is mediated through another place. So for instance, during even before the Imperial Roman period, you have a lot of people that carry ancestry that's similar to what you see in Greece and the Near East. And then when you get particularly Roman urban centers that appear in other parts of Europe, that ancestry becomes pervasive in those areas as well. And it's difficult to say whether that ancestry is coming directly from the Near East or is coming via Rome. Last thing I'm going to talk about is infectious disease. So I hinted at this earlier. So some of the off-target non-human DNA sequences sometimes include infectious pathogens, and so these are detected the same way as the human sequences. You run all your sequences against a panel of genomes of ancient bacteria and viruses, and theoretically we should be able to detect any infectious disease which gets into the bloodstream and for which we have genetic sequences for. Not necessarily every disease is covered, although this is a paper from the Copenhagen Ancient DNA Lab led by Mountain Sikora, looking at the instances of particular diseases through time in Europe. It's difficult to see whether if we find the sequences, it's difficult to say whether they definitely died of the disease or with the disease. Although if it's something like plague during the Black Death, you could be fairly sure. False negative rates are likely to be very high. So a lot of individuals that we sample that did carry a disease, it's likely that they'll test negative for it just because of the vagaries of preservation. So for instance, there was a study of Black Death cemeteries where it was pretty certain that the vast majority of people in the cemeteries in these medieval Black Death cemeteries died of the Black Death, but maybe 1 in 10 tested positive for having Yersinia pestis. So it's likely a high false negative rate. And what's becoming clear is it's likely that the species around genetically that were around in the past, similar species that are around today, actually function differently than in the past. The genomes are slightly different, and it's possible that in many cases they're potentially a different sort of disease, which doesn't bode well for kind of historic diagnoses of epidemics, because it could be that a particular disease that has certain symptoms today had different symptoms in the past because it was genetically different to the disease today, even if though it's the same species. And there may have been diseases in the past that no longer exist, and so we cannot yet detect, although we might be able to detect it with intense methods in the future. That would take a lot of work. The final question, which comes up to me a lot, and I'm sure is on all your minds, is whether there's any viable commercial option for it to get ancient DNA done. And in my view, there are people who say that there is, but I don't think there is yet. And certainly the persistent drops in sequencing makes data generation increasingly economical. And the reason why our lab can generate so many sequences is that the laboratory processes are automated. So the robots do a lot of the laboratory processes, where most of the labs, and we can do kind of process 90 samples at once using those labs, but most labs in this country at least do the laboratory processes by hand, in which case you can only do 12 samples at once. So that having the robots massively is a massive scale up. But the main problem isn't really the lab processes, or even the sequencing, it's the intense data processing analysis, which can often take weeks to happen. It involves quite intense study by individual people. So it's really hard to kind of cost for that and in a systematic way. I mean, there are standard methods for doing certain things, but particularly the ancestry analysis, which is what many people are particularly interested in, for individuals as well as populations, can be quite bespoke and complex, where you're having to bring in different populations and compare them and see which is the best model. And you always have data, more and more data coming out at the same time with populations that may be more applicable. So it's awkward at the moment to set up a commercial operation because it's difficult to guarantee consistent results and cost for those results. So for the time being, at least, research funding is a kind of more stable and greater source of income for ancient DNA labs than a commercial operation would be. I mean, I think that eventually will happen, but because everything's done so bespokely, particularly the bioinformatics with bespoke software where you're writing scripts and having to modify things all the time, it's very difficult to sort of bundle that into a consistent model where you can sign a contract which promises a certain outcome for a certain cost. But again, so up until then, you'll just have to give us samples and let us do it for free instead. But have to prepare for the fact that there will be, as Melina well knows, there will be have to work to our timescales to some extent, and might not necessarily always fit with your timescales. So sorry I've overrun, and thanks. I hope you've managed to hang on in there, and thank you for listening.
Okay, thank you so much. That was really interesting, Tom. I'm just going to pass you over to Sabrina, who's going to do some questions.
Hi, can you hear me okay?
Yep, all
Hi,
good.
so we just have well, thank you for the talk, and we just have a couple of questions to start us off. So what bones should be chosen for the sampling if no teeth, petrous portions, or auditory ossicles are available?
So at the moment, I mean, people are trying different things, and sometimes bones can just work sporadically without being able to predict. But generally speaking, everything apart from the petrous and the teeth and the auditory ossicles have a much lower chance of working. But the ones that do seem to work more often are the phalanges and the talus in some bizarre way. It's a very specific part of the talus. It's just on the neck of the talus. They seem to work a bit more consistently well than any other bone. So in lieu of anything else, those were the ones that I would try to go for. Like I said, it's unlikely they'll work as well as an auditory ossicle or a petrous would, but there's a greater chance of them working than any other bone that people have investigated so far.
Great, thank you. And we also have another question is, what happens to the data produced? Is there a common site where data is shared, or is data kept at the institutional level?
No, there's usually there's a thing called there are different repositories that you can upload data onto, and there's sort of various different kinds, but the main one is the European Nucleotide Archive is what most people upload things on. And technically anyone there's no sort of barriers to access the European Nucleotide Archive. Anyone can download that data, but it is the raw data. So, you know, in theory, it's open to everyone. In practice, it's only open to people who know how to process it, which is a very specific set of people with bioinformatic and biomolecular processes. Like I couldn't process the data from that is available on there. You would need a bioinformatician, someone trained in the methods to do it.
Great, thank you. Are there any other questions? I can't see anything else in the chat, but I saw one. Could you please tell us a bit more about your project? Are you taking samples from everywhere? I think this is the generating
So
whole genome.
generally definitely from Britain and most time periods with a slight, we prefer sort of up until maybe 1000 AD, before 1000 AD, anything before that from Britain. But we're also starting to kind of branch out a little bit into continental Europe as well. So anything from continental Europe between around 1500, sorry, I say about 2000 BC and 1500 AD, we would take samples. I mean, the broad aims of the project is looking at, well, sort of genetic history, broad genetic history over time, but also selection. And for that, you need high quality data, which we're trying to generate, but also large numbers of samples in order to look at it. So the kind of sample size is justified by the fact that you need, like, the larger your sample size, the more powerful your investigation of human evolution, of natural selection can be. But obviously within that, we're naturally generating all this information on relationships and genetic ancestry and sex and things. So as far as possible, we want that to be useful and available to archaeologists, not just the data plonked onto a database, because obviously that's not accessible to most archaeologists, but that we will feed back the results and discuss with regards in a sort of comprehensible way with regards to kind of sex and ancestry and relationships, and then we can talk about what then you want to do that with that as a collaborator, happy to contribute to publications or kind of outreach or whatever you want to use that data for. So, you know, I mean, we have these questions that we have to address because we're a biomedical research institute, and we have to address these questions of human evolution, particularly as it relates to medically relevant variants. But we do have the capacity to do all this other cool stuff that archaeologists and well, me as an archaeologist, are more interested in really than the kind of human evolutionary story.
Great, thank you. That sounds really interesting. Look forward to seeing how it turns out. We had another question as well. Can cremated bone be used to sample a DNA from?
No, we have tried. So just to give you a sense that we run these blanks with all the samples that we run through our laboratory processes, which are just water, just to basically make sure that there's not been any inordinate level of contamination through the lab processes in the water. And we ran it was actually an auditory ossicle, a cremated auditory ossicle in one batch, and essentially it had less DNA in than the water did, the control sample. It was so bad. But I think that someone will eventually get a bit of DNA out of a cremation where maybe the petrus hasn't been burnt as much, and they use a targeted capture array to just really get as much of the human DNA out of there. But it's never going to be I don't think we're ever going to be able to systematically get DNA out of cremations. It just damages the DNA too much for it to survive over even potentially quite short timescales, sadly, because it's always going to leave a big hole in the story of human populations over time, where they go through periods where they're cremating a lot.
Thank you. That makes sense. We also have another question on how many samples from a population is enough. So if you have 90 individuals, how many samples would you be taking?
I mean, in terms of like our own project, you know, in some ways the more the better, because it increases what we can do. In terms of to feel like you've got a representative sample, it depends. I mean, if you've got, say, 10 samples and they're all saying the same thing, then I think you can be reasonably sure that you're getting representative, unless you have some inkling that every single one of those individuals is a first generation migrant. Then it will start to feel like that population you know, their ancestry is very similar. They're deriving their ancestry from the same pool of ancestors. So it'd be very strange unless you've got odd first generation migrants for then the entire cemetery to be different to that. Unless you've got any indications that, again, you've got this structure where you have maybe different funerary rites and perhaps people that carry different ancestries are being treated in different ways. So, I mean, a good example of this is kind of the Chalcolithic to early Bronze Age in Britain, where you have people coming in with new ancestries from continental Europe, um, and they're very archaeologically visible. They're buried in barrows. Most of the dead, or a lot of the dead, aren't burnt. Whereas the population that had been in Britain previously were doing things with the dead that leave no record, and a lot of the dead were cremated. And it looks like this very dramatic and big shift in genetic ancestry in the beginning of the Bronze Age, or the beginning of the Chalcolithic early Bronze Age in Britain. But actually, if that population had carried on sort of living their lives in parallel with these new groups and carrying on cremating, then there's a massive bias towards that incoming group because they're doing things with their dead that actually that leave a record. And we know there has to be a big change eventually, but the mechanism of that change and the time over which that change happens may be much more prolonged. It's just that we can't see one side of the story because they're cremating the dead. So I mean, it's contextually dependent that in cases where you think that there could be people with different ancestries living in quite close proximity, but not interacting with each other biologically so much, then it becomes very difficult to know whether your sample is representative or not. It's sort of, yeah, very much contextually dependent.
Thank you. That makes a lot of sense as well. I'm looking through the chat. I can't see any other questions other than a lot of thank yous for a very interesting talk.
Okay, should we move on then? Yep.
Feel free to email if you have any other questions. I'm happy to answer anything else if anyone has anything else.
All right. Thank you so much, Tom. That was really, really useful and interesting. Just a really quick before we go, we've already set up our next Osteobite. That's going to be on April 23rd, 6:30 again. It's going to be with Dr. Sarah Stark, and she's going to be talking about updates on the Historic England's commission program. So I think we will be sending out links to be able to register for that tomorrow. And just before everybody disappears, if you guys can just put in the chat, we're looking for ideas on other topics and other people you would like to have come and give a talk. So please, can you just add any ideas or on topics or people into the chat just so we can start collecting that. Our newsletter, hopefully everyone's got it. It went out last month. We're going to send out another one probably in the next 2 to 3 months. So we're going to try to get about 4 out every year. At least 3, but we're aiming for 4. We'll see how it goes. But we want sort of our members to kind of help us with this and bring us some content basically. So instead of us just telling you pretty much what we're doing, we kind of want to be able to publicize the things that you guys are doing and the things that you're achieving. So if you're working on any really cool projects or finished projects, let us know. If you've got any students, or if you are a student and you're finishing your MSc or your PhD, again, please let us know so we can celebrate it. Publications as well, so not just published articles, but also if you've completed a monograph, or if you've done a chapter in a book, or even if it's just it's what I mostly do, osteological reports, things like that, anything of interest really, that you kind of like, to get out. I find, especially doing osteological reports, that people don't generally publish those. And so getting hold of them can be quite difficult, especially if you're looking for comparative data on other commercial populations and things like that. So hopefully we're trying to help people kind of get to know where people are doing their research and where they're working. So yeah, so again, I'll send a form out. So please fill that in and send us all your news and projects and celebrate everything that everyone is doing, really.